Hello, everyone, and welcome to the third and final lecture of Chapter 26 here. We are moving along to the final stages of the nephron here. We're going to get through the distal convoluted tubule, and we'll move into the collecting duct or the collecting system, which is technically not part of the nephron. We'll talk about that very briefly. And then the final lecture, part 2 of this third lecture, will be urine transport, the final organs of the urinary tract, and a little bit of histology. So we'll be wrapping this up rather quickly here. So we're going to be focused on part 1, the DCT and the collecting system, along with the hormones here. So let's get into this here. All right, so notice our objectives for the third lecture, part 1. We got these top 3 objectives. Again, a lot of sort of the basics here, and then eventually we'll wrap up with the final urine travel. As usual with the start of the lecture, so this is part 1 of lecture 3. Here's our citations. Of course, we're following along with Pearson's Fundamentals of Anatomy and Physiology here. All right, so we're just moving right along where the previous lecture finished off with, of course, was wrapping up with the nephron loop. As we see, we've just kind of went through this. We know that the thin descending loop is simple squamous, and that's going to allow for the exchange of water, so water being reabsorbed. And then as we get down to the bottom, that's where kind of the solute concentration was getting highly concentrated down there, so lots of salts. And then as we move back up and we start the thin ascending limb, remember that was permeable to sodium, potassium, and chloride. And at the bottom here, it's simple squamous, but once it becomes the thick ascending limb, remember that was active transport of sodium, potassium, and of course, our chloride. But we're focused now on the distal convoluted tubule. So the main point here is we're going to have most of the secretion, so the final adjustments that need to be made. Remember, reabsorption happened primarily here, secretion happens here. And if we think about it, it's filtration, reabsorption, and then our final secretions. Now, again, that doesn't mean zero secretion happens here, and that doesn't mean zero reabsorption occurs here. We're going to talk about those things, but again, the main points are filtration is primarily done at the renal corpuscle. The reabsorption is primarily done at the proximal convoluted tubule, and then the secretion is at the distal convoluted tubule. And what you should really think about the nephron loop is the adjustments of salts and water reabsorption, really. And that's what's going on. So let's get into the distal convoluted tubule. So at this point, only about 15 to 20% of the original filtrate volume actually reaches this. So notice we really reabsorbed a lot of not only the solutes, but we've also reabsorbed a lot of the water as well. So notice, or just remember that out of the 180 liters of urine that ends up starting this process, right? 180 liters gets pushed through the glomerulus. Out of those 180 liters that get pushed through, remember, only 2 liters ends up becoming urine. So a small portion actually reaches the DCT, which should make sense because we're reabsorbing a lot of those components. All right, but the main point of the DCT is it's the site of, notice, selective reabsorption and selective secretion, and note, site of most secretion. So that's the important detail. Tends to be the things that I like to focus on because, of course, this is the big picture stuff because this is likely a lot of the students' first exposure to anatomy and physiology. So, of course, this is a highly detailed course. So the major point here when it comes to the DCT, make sure you know it's selective reabsorption and secretion. Remember when we were talking the renal corpuscle and we talked filtration, remember that was an unselective process. Glomerular filtration was highly unselective. Here at the DCT, now we are making the adjustments because that's where we've moved through most of this nephron. So we got to make sure the final things that are going to form urine should be there. So notice, one, it's the active secretion of ions, acids, drugs, toxins, etc. into the actual tubule, into the tubular fluid there. All right, now notice there is selective reabsorption of, notice, sodium and potassium from tubular fluid. What was the main hormone that we focused on where we talked about exchange of sodium and potassium? And again, at this point, it's one of the main blood pressure hormones. We've talked a lot about it, and then if you're still struggling with this, you should be looking around at the figures, and if you notice, there, of course, is aldosterone. So notice aldosterone stimulates the synthesis and the addition of sodium channels along the DCT, and that's important to note. All right, so sodium potassium exchange and aldosterone sensitive portion of the DCT. So that's the major point. It's under the influence of aldosterone. Okay, aldosterone regulated pump, and that's what the A stands for. Usually the A stands for, oftentimes it stands for active transport, but that's not what this is. This is just saying under the influence of aldosterone. So notice it will increase the sodium in the bloodstream, but of course leads to a reduction of potassium. We've seen this. You need to know that this targets the DCT. I've already mentioned, if you remember, angiotensin 2 targets the PCT. Nothing was discussed about the nephron loop. So notice DCT you need to know, and PCT you really should know these. All right, so this ultimately will lead to an increase in blood volume, which we know that means an increase in blood pressure, but it'll also lead to a decrease in urine volume because wherever sodium goes, we know water follows, right? So sodium's getting reabsorbed into the bloodstream, water is going to follow via osmosis. Just think, wherever sodium goes, we know in our learning that wherever sodium goes, other salts and water are going to tend to follow. Okay, but here we're strictly focused on sodium in exchange for potassium. All right, and then notice for number 3, there is the selective reabsorption of water, and that occurs via ADH. Okay, and after thinking about this for a second, this really should be noting ADH and knowing that it targets, again, the DCT. So I changed that color. And I've also stressed active secretion. So really making sure you're getting the major points down by highlighting those areas there. And that, of course, leaves us with the final ingredient of regulation, which, if you think back to the endocrine system, we talked about calcium regulation. If you remember, parathyroid hormone and calcitonin both targeted the kidneys, but notice now the expectation is that you notice that it targets specifically the DCT of the kidney. So notice calcitonin and PTH responsible for blood calcium regulation, either by stimulating absorption of calcium or stimulating secretion. And if you remember, calcitonin, that's going to end up stimulating secretion, and PTH was the reabsorption of calcium. So make sure you have these down. I didn't put those points down here. So again, calcitonin, the way I said you should remember this is calcium into bone, which means that's reducing blood calcium, and therefore calcium at the calcitonin at the kidney would be secretion, where parathyroid hormone, remember, is stimulating osteoclasts, and at the same time it's going to stimulate reabsorption at the kidneys. So make sure you know those. Just, I kept this very brief, that it's blood calcium regulation, because we've talked about it so much. All right, so a lot of hormones target this area. We have ADH, we have aldosterone, so we're regulating essentially blood. We're essentially regulating our blood volume as well as our salt concentrations, and then calcium regulation. So got to get that down. All right, so secretion at the DCT. So some of the major points to be aware of here when we're going through this. Blood entering at the peritubular capillaries near the DCT may still have undesirable substances. Again, only 15 to 20% of the original filtrate reaches here. But here, remember, we might have some undesirable substances within either the peritubular capillaries, or we might have substances within the actual tubular fluid that we want. So when it comes to secretion, though, that's going to be getting rid of the undesirable substances. So what's going to happen is substances for secretion, they move from the capillaries into the peritubular fluid. So that's what we would end up going from. Here's the capillaries. We would end up going here. Then we would end up moving from peritubular fluids into the peritubular cells, and then peritubular cells into the actual tubular fluid. So notice, if concentrations get too high within the actual tubular fluid, substances will move into the actual tubular fluid. So that's what ends up happening through this regulation, is we're balancing out these fluids here. All right, so that's why all the fluids are being mentioned. So you have to think of it as 3 different fluids. I wouldn't get caught up on this, but just understand, as concentrations get high in one area, if they build up in here, they're going to move into the cells or into the actual different fluids. Okay, so when we talk about secretion at the DCT, remember we have potassium secretion, and that's the counter transport for sodium. We talked about this already, right? We've stressed that that's under the influence of aldosterone. All right. We have H plus secretion. So this one we did not talk about, but I've hinted at on the earlier lecture. All right. So if you remember, H plus secretion occurs when pH falls. So if we have a low pH or an acidic pH, we secrete hydrogen. So that's what you want to add to your notes. When we have an acidic blood, or we have an acidic pH in our body at the kidneys, we're going to secrete H plus ions. By doing that, we're getting rid of hydrogens, and therefore the pH will start to increase. All right. Um, the other main molecule here that we were talking about was ADH. So again, ADH targets the DCT. Now we're going to explain what this actually does. We know at this point, you should know ADH is going to stimulate the reabsorption of water. We're going to go to the cellular level to explain how that occurs. And at the cellular level, ADH is going to actually cause aquaporin production. And that's what I expect students to know. So that's the detail. So ADH, when it reaches these cells, it stimulates the production of aquaporins. Aquaporins get added into these tubular cells here. And now that we have aquaporins, water will be flowing in more readily due to those proteins allowing better water flow versus simple diffusion. Okay, so that's what you need to understand here. So here, K plus secretion is an exchange, counter exchange for sodium. We have H plus secretion when pH is low. Now, on the flip side, just be aware, on the flip side, if our pH was really high, let's say we had a high pH or a basic pH, we would have H plus reabsorption and we would have bicarbonate secretion that ends up occurring there. All right, and they kind of show that here. So notice H plus secretion and HCO3 reabsorption along the DCT and collecting duct. This is how we're going to regulate our pH. It's all related to H plus. When we're secreting H plus, that's to increase pH, which means we will reabsorb this. This is the buffer, and the buffer can also reabsorb H plus ions. On the exact flip side, so you might want to circle this and say this occurs when we have an acidic pH. When we have a basic pH, so maybe off to the side, when we have a basic pH, we are going to secrete HCO3 and we are going to reabsorb H plus when our blood is basic. All right, so a lot to know. We'll be revisiting that, but I'm stressing these points now. And if you didn't get the major point of the DCT, it's the main site of selective secretion, and it's selective secretion based on the presence of hormones. And the major hormones are related to water in the case of ADH. ADH is aquaporin production. We also have regulation of sodium and potassium via aldosterone. Aldosterone, of course, causes sodium reabsorption and potassium excretion. And then we also talked about calcitonin. Calcitonin, of course, is going to reduce blood calcium levels, so calcitonin will cause calcium secretion, while PTH will cause calcium reabsorption. That really summarizes what's going on at the DCT. So a lot to know, but stick to the main points. What ingredients are being exchanged? All right. Again, don't get too caught up on this. This is just basically telling you the way this works is as things build up in here, that's going to help with exchange. But of course, as these build up, they're either going to get reabsorbed by the bloodstream, which means it's going to get moved right along, or it goes into the tubular fluid, which of course the fluid in the tubular is all in the tubes is also moving. Don't get caught up on this point here. It's not a major thing to understand. The main point that I'm trying to stress with this bullet point is pointing out that as these build up, that's going to influence exchange and what ingredients go where. Major points are to focus on the hormones and that this is the site of secretion. And we've finally made it to the collecting ducts. Now, you have to keep in mind the collecting ducts are not considered part of the nephron, and the reason being is when we define the collecting duct, it's what receives fluid from many nephrons. The fact that multiple nephrons are connected to this, you can't single it out and say, hey, it's part of a nephron because it's literally multiple nephrons joined to it. So it's called the collecting ducts, or it's part of the collecting system. And so that's why it's not considered part of the nephron. Very easy for me to ask which of the following is not part of the actual nephron. And, renal corpuscle, PCT, nephron loop, DCT, and then I just list collecting duct, and you should know it's not technically part of the nephron. Again, it's a technicality. It's part of the anatomical description side. Is it really that big of a detail for me? No, but I would expect you to know it's something that I will actually quiz on. But in the grand scheme of things, when you're describing urine formation, if you didn't know that, would that be a huge problem? Probably not the biggest deal in the world. Instead, you should definitely know that, hey, it starts off as our typical simple cuboidal. I think I kind of failed to really point that out, that the DCT is also simple cuboidal. Hopefully you learned that from the lab lectures and so forth, but you should know the DCT is also simple cuboidal. Once we get into the collecting duct, you should know it's simple cuboidal, and then it becomes simple columnar once we reach the papillary duct. So it starts to widen out, and as it does that, you should know it's changing. So if I ask anything about the collecting duct, the type of epithelium, the answer I expect for my students specifically is starts as simple cuboidal and becomes simple columnar. That should be your answer, and that's what I would expect you to do. If you're taking a different course and you're not in mine, you might want to ask the instructor. They have some sort of similar question and some sort of expectation. Now, interestingly, with the collecting duct, it's pretty similar to the DCT, but again, DCT was specialized for calcitonin and PTH. This, the collecting duct, still going to be target of aldosterone and still going to be a target for ADH. Okay, so notice again, this should be, you should know this. Aldosterone is sodium reabsorption for potassium exchange. Okay, potassium secretion, notice reabsorption and secretion. Those terms should now have a lot more meaning in comparison to when we began this unit, or definitely in comparison to when we began this semester. ADH, as we know, is water reabsorption. It leads to more concentrated urine, but less urine ends up forming. So notice here, this is under the influence of ADH. We see they do a pretty good job. Notice they're showing you water reabsorption. They're showing you sodium chloride exchange here. They're showing you ADH targeting the targeting the DCT. They're showing you ADH targeting the collecting duct. Now, basically, where they're showing this water reabsorption, that would be where those proteins, those aquaporins, were added. And so that's why water is able to flow back into the body. And you can see at the renal medulla, that would end up causing the renal medulla to become less concentrated with salts because water is flowing into there. But eventually, that's likely to go into either the vasa recta or into the peritubular capillary. So it'll become part of our actual bloodstream. All right, so nothing new here. You should know the collecting duct is the final site of urine flow before exiting into the minor calyx, right? All right, the other hormone that you should know, and we didn't really talk about this, but remember the counter to ADH and aldosterone is the diuretics, right? So natriuretic peptides and diuretics in general, but we're going to just stick with the natriuretic peptides. But if you've ever heard the term diuretics, this is what diuretics do. They help form dilute urine and create a constant urine flow. And by doing that, of course, you're reducing blood volume, which is thought to help regulate blood pressure. So in that case, you're blocking basically ADH formation. Now, note that in the collecting duct, there might be some urea reabsorption. I don't stress this point. It's not something that's super relevant to me. Obviously, we're forming urea. That's the goal, and most of the bad stuff is going to be urea. That's the main waste that we're getting rid of. The fact that we reabsorb some here, it's more of an FYI for you. But still note, the final thing is the collecting ducts, the natriuretic peptides can do this, and what they do is they block ADH formation. And so instead of this water coming in, notice you just have a straight flow of water, and so you have a larger volume of urine, but it's going to be much more diluted. And so notice the permeabilities and the osmotic concentration of urine without. So in this case, you don't have those aquaporins, and so you're going to form large volume, but it's not going to be concentrated. And the final major point to really get down here is this is the final site of pH adjustment, and that's going to happen by either secreting or reabsorbing the bicarbonate ion and the H+. And so when it comes to adjusting it, we're either going to secrete bicarbonate and reabsorb H+, or we're going to secrete H+ and reabsorb bicarbonate ion. Now, let's just say our body's pH was high. So we have an alkaline sort of pH, meaning a basic pH in our blood. What would we do to decrease the pH? I.e., we need to increase the acidity because we're alkaline. So how do we increase the acidity, or how do we decrease the pH? So think about that. What would we secrete and what would we reabsorb if we're trying to increase the acidity in the bloodstream and thereby reducing the pH? These are the sort of questions, because of course on the flip side of things, I would say, how do we increase pH? So here we're focused on the decrease. So pause it if you want to think of this for a second. But the way we would decrease our pH or cause a pH decline is we would secrete the bicarbonate ions, because this is the buffer, right? And then we would reabsorb or pump H+ back into the peritubular fluid, which would end up going into the peritubular capillaries or vasa recta, meaning it would enter into our bloodstream. And essentially what we're doing here is at the kidney, we're getting rid of the buffer and we're picking up H+. And by absorbing more H+, that makes it more acidic. which reduces the pH. And so that's what you should end up walking away kind of from this whole concept here. So where I'm going to be stressing this several times throughout this last unit, get this concept down on how pH regulation occurs. All right, and so here in your book, you do have figure 26.16 that really summarizes details. So notice they talk about the simple squamous epithelium, they talk about simple cuboidal with microvilli, they talk about here at the descending limb being simple squamous versus the thick limb being simple cuboidal. Here, distal convoluted tubule. Notice it does not have the microvilli, so that's how you kind of tell the differences. And we'll go to that and talk about that when we get to the histology in just a second. The other big figure, so this is actually table 26-1. The figure that overviews all the events is right here, and you can see what's going on here, and they try to relate it to the osmolarity along the way. It's not something that I get too caught up on relating this to the blood concentration, so don't worry too much about those numbers, but if you want to see what's going on, this is a really great figure, and notice as this goes down, notice the vasa recta is pulling things away. So it's an excellent figure to just kind of overview the things here, but I kind of did my own summary. So I would encourage you to try to make sense of this. But again, they're going to get into more details that than you need to know. But sometimes going a step further really can help make a lot more sense. And so you can see they're talking about the collecting duct and DCT. And so really great figure. I can't stress reviewing it enough. All right, but let's go on to the summary of events here. And so here they're showing you how the cortex, notice the concentration of basically salt, the salt concentration. As we move to the medulla, just note that salt concentrations are building up there. That was the major point. All right, but summary of events. Urine formation, you should know, begins via filtration. And filtration is strictly occurring at the glomerulus or the renal corpuscle. It's occurring at the glomerulus capillaries or within the renal corpuscle. That's what you needed to know, and that's what we're seeing right here. So site of filtration. With that, you should know filtration is an unselective process that relies on the net filtration pressure. And during that process, approximately 180 liters of filtrate are going to form. But as we know, by the time we reach the DCT, 15 to 20% of the original filtrate has already been reabsorbed, which notice this is why we only are going to end up with 2 liters of urine, and we're only going to form about 178 liters of that filtrate actually gets reabsorbed. So only 2 liters of urine on average are formed from individual to individual. And again, this is the average. It's not the specific number for every person. Then we moved along and we get to the PCT. So the main points of the PCT was it's the main site of reabsorption, 99% of our good nutrients. The thing that we said that it relies on was the sodium exchange, because sodium drives the ion exchange, right? So sodium gets in, that helps move some of the ions. Some of the other ions get actively transported with or symported, right? That was the co-transport or symported together. As that salts build up, that causes the movement of water by osmosis, and then after osmosis, then it's passive flow of ions. That's the detailed side of things. But if you just focus really on this bullet point, you're probably good. Notice angiotensin 2 helps with the enhancement of sodium absorption there. All right, so that's the PCT. Main site of reabsorption, most of our good stuff, relies on sodium exchange and angiotensin 2. Nephron loop, that was water and salt reabsorption during the thin ascending limb. That's where water gets reabsorbed in that simple squamous epithelium. At the loop, it's a balance, so there really isn't much exchange going on. But as we move up the thin ascending limb, you should know it was sodium, chloride, and actually potassium. They're not showing potassium on this figure. But notice when we get to the thick ascending limb, this is active transport of the sodium, potassium, and chloride. Basically, it's the movements of salts and water. Then we get to the DCT, that's the main site of secretion, and that secretion is going to happen by the various hormones that we've already discussed, but primarily ADH, aldosterone, and then, of course, we've talked about calcitonin and PTH. So don't forget, calcium regulation happens at the DCT. Once we reach the collecting duct, it's strictly the ADH and the aldosterone, but don't forget natriuretic peptides. So you might want to add natriuretic peptides to the list here, which I have actually done for you here. And then don't forget, our pH balance occurs along the PCT, DCT, and collecting duct, i.e. it doesn't occur during filtration, and it really doesn't occur during this area down here, although we are technically losing some chloride, but that's not really the balance. It's hydrogen. So overall, this really should summarize your main points. Now, it is a tough process, but really focus on the keywords like filtration, reabsorption, secretion. Okay, that summarizes some of the major points right there. Water reabsorption at the thin limb, sodium, potassium, and chloride at the ascending limb, pH balance. A lot of the major points are right here in the big picture stuff. So if you know the big picture stuff, I would say you probably could do around a 70% on my exams, knowing these big picture details. All right, the final concept that we need to get through, since we are done with the nephron and all of its parts, really is looking at the histology of the overall nephron. And so you can see several figures here, and if you notice, when you see these sort of structures here, these all tell you that this is the glomerulus. In fact, this is the actual capsule, and then this is the knot, this is the glomerulus itself. So sometimes I'll circle the whole thing and ask, what are all these structures down there? And you would need to know that those are actually considered the renal corpuscle. So what is this entire structure? All right. Otherwise, I can literally point to the center and ask what that is. In the center, that is strictly the glomerulus itself. And you should know, I usually do follow-up questions where I would ask, what type of epithelium? This is simple squamous in here. So same thing, an image like this, what is the structure? I circle the whole thing. It's the renal corpuscle. Otherwise, I point an arrow and say, what is this red structure that the arrow is pointing to? That's the glomerulus, simple squamous. Similarly to arrows pointing to the outside, you should know this is the glomerular capsule on the outside, which is still simple squamous. All right, and that really summarizes the renal corpuscle, what its look is. That is it. You need to be able to get that down. All right, so the glomerulus should be obvious, simple squamous. The whole thing's the renal corpuscle. All right, need to know it. And so glomerulus, glomerular capsule, renal corpuscle. All right. Now, the PCT and DCT, I will give credit for either. Technically, the PCT has the microvilli and the DCT does not, and that's how you would tell the difference. When you're looking at these figures, most of them are a little bit hazy, but if you look here, these ones have, if you notice, PCT, they're a little bit thicker, and you can see the little lines running through it. All right, if you look here, collecting tubules, I wouldn't be able to tell the difference between the collecting tubules versus the actual DCT. That one I'm not too familiar with, but either way, if you see simple cuboidal epithelium, I'm looking for PCT or DCT, and I'll give you credit for either of those. Now, in this case, the follow-up question that I usually ask is, what region of the kidney would this be? Since you're seeing renal corpuscles, you know that this is a histology image of the cortex. This is in the cortex. Where here, notice, we don't see any renal corpuscles. All right, so this is likely the medulla. All right, and the reason I know it's the medulla is there's no renal corpuscles. And so this is where you would have, definitely this case, you would need to know, hey, this is, I would give credit for a lot of things. I would give credit for the thick ascending limb, the thick descending limb, the PCT or the DCT. But here, when we have things that are simple squamous, you would need to know those are the thin limbs. Usually I'm not that strict on it. You can see there's blood vessels, but really the main point of this is, can you identify the that this area is going to be either a PCT or DCT, and then do you know that this is part of the medulla? All right, but that's really going to clear this out. So top images were the renal cortex, the bottom image would be the renal medulla. Simply putting this on the exam saying, hey, what is this a histology image of, and be specific, you would need to know renal medulla. All right, but at this point, this gets us through those top 4 group note review questions. We have the bottom ones remaining, so we got 5 more to get through. That'll be covered in part 2, but thankfully we are done at this point. We are through the entire nephron and all of the kind of microscopic cellular components of this. We're now going to move backwards and kind of look at the macroscopic remainder of this, getting through the ureters, getting through the bladder, and of course the urethra. So we're wrapping up pretty quickly here. As usual, thank you all for listening. If you have questions, make sure you get in contact with me some way. The next part 2 of lecture number 3, is the last part of chapter 26 here. So we'll be finishing that up soon. Thank you all for listening. Have a great rest of the day, and I'll see you back at the final part. Take care.